Welcome to ANA Investigates, a monthly podcast featuring the latest in neurology and neuroscience, brought to you by the American Neurological Association. I'm your host, Adeline Goss. Today, in recognition of Alzheimer's and Brain Awareness Month, we'll hear from Dr. David Holtzman, professor and chair of neurology at Washington University in St. Louis. Dr. Holtzman's work has helped to clarify the relationship between ApoE4 and Alzheimer's disease. He's also working to develop CSF biomarkers for Alzheimer's, and he's worked on the development of anti-amyloid beta and anti-tau antibodies that are now being evaluated through clinical trials. ANA's education manager, Jen Hurley, spoke with Dr. Holtzman about the current state of research in biomarkers for Alzheimer's disease. Here's Jen.
Welcome, Dr. Holtzman. Thanks so much for being here today.
Thanks so much. It's a pleasure to be on.
June is Alzheimer and Brain Awareness Month. I think it's a moment to think about where we are in our understanding and treatment of this disease and where we're headed. So I'd like to start with the diagnosis of Alzheimer's. You've worked a lot on biomarkers, and I wonder if you can tell us a bit about the most promising biomarkers for Alzheimer's disease at this point in time.
I think it's probably worth defining biomarker briefly. So it's a measurable substance in an organism whose presence is indicative of some phenomena, such as some aspect of a disease. And the reason that biomarkers are important for Alzheimer's disease is that there are a number of different diseases that can cause someone to become demented. Alzheimer's disease is the most common cause of dementia in people over 65. However, since there are other causes, one cannot be 100% sure that somebody has Alzheimer's disease only based on the clinical abnormalities that someone presents with. So in this regard, the most promising biomarkers for Alzheimer's disease at this time are the markers that can detect the presence of amyloid deposition, one of the main pathologies of the disease, and or the pathology of the tau protein. There are now ways to use PET scans where one can image the presence of amyloid in the brain of a living person, whether or not they are symptomatic or not symptomatic. And those tests work very well. There are also ways to use PET scans now where one can image the tau protein and its accumulation in Alzheimer's disease. And those tests are also work very effectively to see that pathology. One can also visualize that pathology by looking at the spinal fluid, where one can determine that somebody has amyloid deposition with certain measurements in the spinal fluid, including amyloid beta 42 levels. And also one can see evidence of amyloid-induced tau pathology by measuring different forms of tau in the CSF.
So I understand that those are some of the established biomarkers of amyloid and tau pathology. Are there any other promising biomarkers that are still in the research stage?
Sure. So for example, there have been several papers published demonstrating that simply by measuring the amyloid elements of the amyloid protein called A beta 40 or 42 in the blood, that if done in the proper fashion, can actually tell you whether or not you have amyloid in the brain or not. Similarly, there are plasma levels of forms of phosphorylated tau that have recently been published or discussed at meetings, where those measurements also can indicate whether you have amyloid in the brain. And I suspect that going forward with research, as these tests become validated, it may be that one won't need to do as invasive of a screen, such as a PET scan, which requires radioactivity, in order to make a diagnosis and get people on the right therapy. But there's a number of other markers, biomarkers, that I think will be also useful that are still in development. So for example, one of the major pathologies that we think is important in Alzheimer's disease, other than amyloid and tau, is the inflammatory response. And it would be really helpful if the field can develop fluid biomarkers, whether they be blood or CSF, as well as imaging biomarkers that better identify the inflammatory response that's occurring in an individual person. Further, there are markers of real degeneration in the brain that have been published on recently. One of them is called neurofilament light chain that can be measured both in the spinal fluid as well as in the blood. And further validation that that kind of a degeneration marker is not only useful, but responds to a therapy. That will also be a very helpful point in the field, so that as treatments are developed and they're approved, you could actually potentially follow in an individual person whether the treatment is actually not just affecting the underlying pathology, but it's slowing the degeneration as measured by a marker like NFL.
What promise does this all hold for future patients, and how do you see these advances making their way to the bedside?
I think there is sort of an immediate need and use versus what I think will be the more important longer term use. So in the immediate term, there are some patients when we see them and they're very mildly impaired, where it's unclear whether they're developing Alzheimer's disease or another disease of some type. And the use of these markers, such as the amyloid or tau imaging or CSF markers, can help one determine whether someone really has Alzheimer's or another disease. And that can help for a variety of things, such as planning, education, and potentially some of the current treatments available, such as cholinesterase inhibitors or other drugs. I think in the longer run, however, markers such as this are going to be really important once there is a treatment that actually is trying to directly affect the underlying Alzheimer's pathology, because you'll really want the way that current trials of promising disease-modifying agents are being done is that the different groups that are doing these studies are identifying with biomarkers who really has amyloid or tau pathology or both, and then enrolling those types of patients in their trials, so that ultimately one will need to know if a drug is approved that's in trials now, there'll be a need to identify who really should get the treatment.
What are some of the other areas of Alzheimer's research you see as holding promise?
One of the things that's really important, I think, that we've learned over the last 20, 25 years is that the pathology, the underlying Alzheimer's disease pathology that ultimately leads to cognitive decline and dementia begins in the brain about approximately 20 years before the symptoms develop. And this pathology slowly accumulates with amyloidosis and tau accumulation and other changes over a long period of time while people are still clinically normal. That's an area where the use of the biomarkers already mentioned is being worked on extensively, because ultimately, if you could identify people in that very long, what we call preclinical window of disease, one could potentially start treating people at that point as identified by biomarkers to hopefully delay or prevent the actual onset of what we know of as Alzheimer's disease. And this would be analogous to what occurs now, for example, with either colon cancer or heart disease, where based on guidelines, you hit a certain age. In the case of colon cancer, everybody's supposed to get colonoscopy every 10 years starting at age 50, so that you can really, if you did have cancer, could be detected and prevented from causing a big problem. So in the case of Alzheimer's disease, I could imagine that once people hit a certain age, they start getting biomarkers assessed. And if some of them are, if they're negative, maybe they would then just get screened again in 10 more years. But if some of them are positive, depending on the development of therapies and how that goes in the future, you might start getting specific treatments started very early to really delay or hopefully even prevent the disease.
Are there any trials ongoing now that are using biomarkers to screen patients who have preclinical Alzheimer's disease?
Yes, in the field, a number of years ago, the first treatment trial of preclinical Alzheimer's disease began, called the A4 trial. And in that trial, individuals who are 65 years and older who are cognitively normal were screened for whether they had amyloid in the brain with either an amyloid PET scan or CSF testing. And only if they were positive were they enrolled in the trial. And that particular trial was testing an anti-amyloid treatment. But subsequently, there are additional trials that will begin. One of them is called A3 and one of them is called A5. So while we don't yet have the results from A4, it's a 4.5-year study. This is the way the field is going now to try to develop treatments that will actually delay the onset of disease.
This is really encouraging to hear. So what are some of the new approaches to treating Alzheimer's that you're most excited about right now?
Probably one of the hottest topics that has not been as explored, other than trying to focus on amyloid and tau, is the inflammatory response. And we now know that many of the newly identified risk factors, genetic risk factors for Alzheimer's disease, are genes that are expressed very highly in the brain in microglial cells, the innate immune cells of the brain. And I think trying to better understand how microglia and the genes that are expressed in them that are risk factors are linked with the disease to ultimately not only understand pathogenesis, but hopefully to develop targeted therapies is, I think, a really hot area right now, and something that hopefully more and more people will work on and understand.
So I'd like to pivot to ask, what are your recommendations for someone just starting out who'd like to focus their research on Alzheimer's disease?
Yeah, I think for physician scientists who make up much of the ANA, there are many possible things that would be interesting to work on going forward. And because Alzheimer's disease is not just one, it's not like studying one particular protein or one part of the brain. It really offers an opportunity to study several things. For example, if one's main interest is in studying systems neuroscience or how the brain really works, there are a variety of interesting technologies and ways to study normal brain function in the context of a disease like Alzheimer's disease that are very exciting to work on. If you're more interested in the underlying cell biology or biochemistry of Alzheimer's disease, there are many exciting things to work on there. So I think it really depends. There's still a lot to work on, and we still have no treatment for one of the most devastating diseases in humans. So I think what one ultimately works on would have to do, one, do you have an interest in this general topic of this disease? And two, scientifically, is there something that really turns you on to work on in regard to the type of science you want to do, whether it's in humans, in animals, in cells? So there's lots of possibilities.
Well, Dr. Holtzman, it's been an honor speaking with you today on ANA Investigates. Thanks so much for being with us.
Thank you, Jen.
That was Jen Hurley, ANA's education manager, interviewing Dr. David Holtzman, professor and chair of neurology at Washington University in St. Louis. That's it for today's show. You can hear past episodes and submit your own ideas for the podcast under the Education tab at myana.org. For ANA Investigates, I'm Adeline Goss.
This has been an episode of ANA Investigates, the official podcast of the American Neurological Association. Thanks for joining us for this episode. If you're not already a member of the ANA, please visit myana.org to learn more about the benefits available to ANA members, including access to an array of educational programs and resources, like online CME courses, webinars, the ANA's journals, the Annals of Neurology and the Annals of Clinical and Translational Neurology, and more. Dr. Adeline Goss is our host. Our producers are Drs. Romer Giucaden, Adeline Goss, Michelle Johansson, Clifton Gooch, Danny Bega, Megan Ritchie, and Jim Sigler. This is your editor, Jen Hurley. Copyright, the American Neurological Association.